So we were discussing the acid-base balance in our body, which occurs through three mechanisms, which were renal mechanism, the respiratory mechanism, and here the first one, which is the acid-base balance by a buffer system. What is buffer system? Acid-base buffer system consists of a weak acid and a base. Weak acid is a protonated substance, while weak base is a unprotonated substance. So this will making the acid-base buffer system. Now this buffer system resists the change in the pH and prevent the body or a cell or a fluid from the alkalosis or acidosis. So this weak acid and a base combination, which is acid-base buffer system, it combines with the H plus ions. These H plus ions, this system that is weak acid or base, when combined with the H plus ion, this will negotiate its effect. So we have three types of the buffer system, that is bicarbonate buffer system, phosphate buffer system, and the third one is a protein buffer system. The number one is the bicarbonate buffer system. Suppose we have a fluid like ECl. Whenever there is a altered or a change in pH, like when we add HCl, which is an acid, it will be broken down into the H plus ions and Cl minus. So this H plus ion will decrease the pH of the solution or a fluid, obviously. So this buffer system, that is bicarbonate buffer system, when added to that fluid or a solution, that is NaHCO3, which is a bicarbonate buffer. So this NaHCO3 will alter the pH, that is not going to decrease. So this NaHCO3 is broken down into the HCO3 and Na plus ions. So this HCO3 and minus ions, when this is broken, this combines with the H plus ions. So these combines and they will form the H2CO3, which is a weak acid. So this HCl, overall equation is like this. This will form Na will combine with the Cl, and this bicarbonate HCO3 portion is combining to the H plus ions, and they will forming the H2CO3. So two products are formed, that is NaCl and H2CO3. This carbonic acid, which is H2CO3 is broken down into H2O, that is water, and CO2. This H2O and CO2 are not too much harmful like CO2. CO2 in excess is harmful, but this is countered, or the mechanism, that is respiratory mechanism, will eliminate this excessive carbon dioxide. So this is how the bicarbonate buffer system acts in a decreased pH, whenever there is a pH decrease. So in the same way, this bicarbonate buffer system also acts when there is a increase in the pH. Whenever there is an increase in pH, the bicarbonate buffer is added. Second one here is the phosphate buffer system. Phosphate buffer system, like bicarbonate buffer system, also consists of a weak acid and a base. It is playing its role in the ICF and ECF, which is containing the acids or bases, and so this is countered by phosphate buffer system. So this phosphate buffer system is playing a very major role in the RBCs. The RBCs, it is maintaining the pH. So this buffer system, when acts like when HCl is added in a fluid, it is it is strong acid. When HCl is added to the solution containing a buffer, that is Na2HPO4, this phosphate buffer will convert this strong acid into the weak acid, which is Na2HPO4 or NaH2PO4. This is the weak acid, and NaCl is formed. So this is how phosphate buffer is counteracting the strong acids. Now when we add a strong base like NaOH, phosphate buffer will act NaH2PO4, which will convert this strong base into the weak base, which is forming NaHPO4. This is actually converting the strong acids into the weak acid and the strong bases into the weak bases, and not affecting the ICF, ECF and the cells to change or alter its pH. This is how the phosphate buffer system acts. Next one is the protein buffer system. The third one here is the protein buffer system. Protein buffer system is present in the blood, in the plasma and in the erythrocytes. In plasma, it is present in the form of carboxyl group or some part of the terminal amino acid group like glycine, and it is present in the plasma in this form and maintaining the pH. In second one is the erythrocytes. In erythrocytes, the major protein is present is hemoglobin. Hemoglobin is 6 times more stronger protein buffer system, maintaining the pH than the proteins buffer system present in the plasma. So this hemoglobin is carrying oxygen also, but it is acting as a protein buffer system. So deoxygenated hemoglobin is more powerful buffer than oxygenated hemoglobin. Actually, the hemoglobin in the deoxygenated capillaries, the capillaries containing the blood which is deoxygenated, where the H plus ions are present, this deoxygenated hemoglobin combines with these H plus ions until more and more carbon dioxides are entering inside these capillaries. So this is maintaining the concentration of H plus ions in the blood present in the capillaries. So this hemoglobin is playing very important role in maintaining the pH in the blood. This one was all about the buffer systems. Types of the buffer systems of the acid base. Number one is the bicarbonate, second one phosphate, and the third one we have discussed the protein buffer system. The other regulation mechanism, which is through respiratory mechanism or through renal mechanism. Through renal mechanism, the H plus ions are excreted out and the pH is maintained, and likewise the HCO3 minus ions are preserved in the body. This is how the renal mechanism acts. So here is the second one, respiratory mechanism. Respiratory mechanism acts through eliminating the CO2, which is a source for the acid, which is weak acid actually, a carbonic acid. H2O plus CO2 gives H2CO3. This is a carbonic acid. So as a result of the breakdown of the H2CO3, CO2 is eliminated. This CO2 is eliminated off from the body by a respiratory mechanism by ventilation. This is how this CO2 is eliminated off and the pH of the system of our body is maintained. So these are the three systems, that is regulation through buffer system, through respiratory mechanism and through renal mechanism. This is how the pH of the body is maintained. Now we will discuss the applied physiologies like acidosis, alkalosis, respiratory acidosis, respiratory alkalosis. All these we will discuss in the next lecture. This is all about the basic mechanism which are involved in the maintenance of the acid-base balance.